It is an undeniable fact that inflammation is crucial for host defense. But inflammation is not always beneficial to the host and can lead to serious tissue damage and organ dysfunction. In this study, we sought to investigate regulatory mechanisms that allow tissues to protect themselves from unwanted inflammation in response to sterile tissue damage, such as the local death of a few stromal cells. The use of state-of-the-art intravital imaging techniques allowed us to directly look at the very first steps of an inflammatory response in different organs in a live but anesthetized mouse. To generate local damage in these tissues, we used a short laser pulse to induce the necrotic death of several stromal cells. As part of the inflammatory response to this injury, a highly migratory population of innate immune cells, called neutrophils, was recruited into the tissue and directly targeted the damaged area. Upon contact with the debris, these leading neutrophils became activated and underwent inflammatory cell death, which led to the recruitment of more and more activated neutrophils and the formation of dense swarms at the site of the damage. This swarming activity led to collateral damage of the tissue and further extended the local disruption of tissue architecture produced by the initial insult. To see if very limited tissue damage would lead to the same inflammatory response, we downscaled the laser injury to just a single cell. At first, neutrophils were still recruited in response to the injury, but instead of directly targeting the damage, they kept wandering through the tissue without inducing actual inflammation. To understand why neutrophils were not swarming at small lesions, we took a closer look at the composition of immune cells in the tissue at the time of injury induction. We found that in mice, most internal organs like the brain, muscles, and various interstitial and organ lining tissues contained a non-migratory population of resident tissue phagocytes called macrophages. These cells were uniformly distributed, which allowed them to optimally survey the whole tissue area by continuously sampling the extracellular space between them. When we induced a small laser injury, these tissue-resident cells rapidly sensed the damage and extended long membrane processes to cover and sequester the debris before neutrophils entered the tissue, a process we termed cloaking. This cloaking blocked the attracted neutrophils from making direct contact with the debris and becoming activated, which in turn prevented swarming, inflammation, and collateral tissue injury. When we removed resident macrophages from the tissue or interfered with their ability to cloak, neutrophils were again able to reach the lesion, become activated, and induce swarming. This showed that the lack of cloaking not only allowed neutrophil-driven inflammation at smallest lesions, but also affected healthy tissue by causing collateral damage. By sequestering pro-inflammatory foci through cloaking, resident tissue macrophages were obviously able to interfere with the initiation of neutrophil activation and subsequent swarming, thereby disrupting potential tissue-damaging inflammatory responses at the earliest possible stage. When we used drug treatments that depleted the resident macrophages, active tissues such as the diaphragm now showed evidence of tissue destruction by neutrophils that was not seen in normal animals with intact macrophage populations. Our study therefore reveals the existence of a previously unknown cloaking response of tissue macrophages that constitutes a novel checkpoint actively preventing steady-state inflammatory damage to diverse organs throughout the body.